In this lecture, we're going to talk about nervous systems and sense organs in animals. Well, let's first talk about what happens in a nervous system. We're talking about the movement of electrochemical signals along and between cell membranes. So, for example, you receive information about the external environment if you hear a phone ring. Well, that information is transferred to your brain, which processes that information and then sends another signal, electrochemical signal, to your muscles to affect some behavior. Now let's talk about where these electrochemical signals are being transferred. The large structure involved is called a nerve, but nerves are actually bundles of neurons bundled together in connective tissue. So these individual purple cells are the neurons. That's another name. A neuron is another name for a nerve cell. And the entire bundle here of neurons is generally referred to as a nerve. So let's break down a neuron into its component parts. First, we have the dendrites, which receive information from either a sense organ or a neighboring neuron. The dendrites are connected to the nucleated cell body, and this is where, as the name implies, the nucleus is located. But then the signal is propagated down the length of the longest part of the neuron, which is called the axon. There's a very long fiber in which we see the propagation of an action potential. This is the electrochemical signal that is running the length of the neuron. And axons sometimes are not covered by a myelin sheath, but sometimes they are covered by this insulating structure called a myelin sheath. It surrounds and insulates the axon. It's seen in vertebrates, and some invertebrates also have myelin sheaths. This structure, this myelin sheath, is actually produced by other cells, which are called glial cells. The signal of a neuron ends up at structures called terminals or synaptic knobs, where this information has to be transferred to another cell, either, again, another neuron, or in this case, a muscle, to cause that muscle to activate. Well, to understand how this electrochemical signal is transferred along the axon, we have to understand the difference between a nerve action potential and its resting membrane potential. When a neuron is not signaling anything, it's said to be at resting potential. So at rest, a neuron has more chlorine and sodium on the outside of the cell membrane, but on the inside we have a larger concentration of large impermeable, negatively charged anions and potassium, K+. So at its resting membrane potential, there's a slight relative negative charge on the inside of the membrane as seen in this middle figure. Now for a signal to propagate down the lengths of the axon, going from the cell body down to the terminals or synaptic knobs, we first have sodium flowing into channels into the interior of the axon. That causes an immediate depolarization. So it swaps the inside from a negative charge to a positive charge. As the sodium flows into the axon, running the length of the axon, we see a subsequent change also in the potassium. Potassium, which was at relatively high concentrations inside the axon, now flows to the outside of the axon. And see, what this does is it reestablishes a slight negative charge on the inside of the axon. And you can see in this figure here how this sodium is flowing into these sodium channels all the way down the length of the axon. It basically acts like a domino effect, going from the cell body region down to the synaptic knobs in a nice wave. But at the end of the action potential, now we've got the sodium and the potassium on the wrong side in the wrong concentrations of the axon's membrane. So to reestablish and get us back to a nice resting membrane potential, we have what's called the activation of the sodium-potassium exchange pump that pumps the potassium inside and the sodium back outside. and this just makes it possible for subsequent action potentials to occur. Now, how fast action potentials occur in animals varies widely among different animals. So it can be as slow in anemones of 0.1 meters per second to some mammalian neurons in which it can travel 120 meters in a second. Well, some of the things that will speed up nerve action potentials is to have a larger diameter axon. The bigger the axon, the faster the speed. This has been studied in the giant axons of squid. They have such large neurons that the axon is actually visible without the aid of a microscope, as seen here. So another thing that you can do to speed up the action potential is to have a myelated neuron. These are also faster, because instead of this steady flow as seen in A here of the sodium flowing in to the axon, propagating down the length of the axon, now you see the electrochemical signal jumping across these insulated regions covered by the myelin sheath. And so it's just a much faster process by leapfrogging these large sections. And again, this is mainly seen in vertebrates, but some invertebrates have also evolved myelin sheaths. And finally, the third way that the nerve action potential can be sped up is temperature. We talked about in an earlier lecture why it's important for animals to maintain a relatively high body temperature, that it's just more efficient for chemical reactions. And again, one of those reactions is the nerve action potential. It's just faster when the neurons are warm. Now, once the nerve action potential runs the length of the axon, then the signal has to be transferred to another neighboring cell. And it does this at the end of the neuron, which is called the terminal or the synaptic knobs. And there are two ways this information can be transferred to the next cell. One way is using a direct transfer of electrical information, and this is called an electrical synapse. This is very fast. It can occur very quickly. It appears to be very rare in animals. Not many animals have this capability. Instead, most animals show what is called a chemical synapse, where there are vesicles in the synaptic knobs that merge with the synaptic knob membrane and release chemicals that go across this synapse, this gap here, and then flow through channels in the neighboring cell membrane of the other cell to chemically transfer this information. These chemicals are called neurotransmitters. And while this is a slower form of transmission of electrochemical signals, it appears to be more common in the evolution of animals. Let's now talk about the evolution of nervous system diversity in animals. Some of the simplest animals we'll discuss in the semester include things like cnidarians and ctenophores, and they have what is called a loose nerve net. It's the simplest of all possible nervous systems. There's an interconnected network of neurons to sensory structures, but there is no centralized controlling structure or no brain. So it's just this loose network. Most animals, however, have a bilateral nervous system, where nervous tissue is subdivided into two parts, the peripheral nervous system and the central nervous system. The peripheral nervous system can be further subdivided into afferent neurons, which could also be called sensory neurons, because these are receiving information about either the interior or the exterior environment. And these receivers of information are called the receptors. And they help to tell the body something about its internal or external conditions. Information from the afferent neurons is then taken to the central nervous system. Once the central nervous system processes this information, then it sends a signal back through the efferent neurons or motor neurons. These connect to effectors, muscles, glands, so they tell the body how to respond to the information that was received from the sensory neurons in the afferent nervous system. But again, central to this in the peripheral nervous system, receiving the information and then delivering the information back to the peripheral nervous system, is the central coordinating central nervous system. So this includes the brain and the nervous cord, but then also involves very small neurons that connect the central nervous system to the peripheral nervous system, and these are called interneurons. One of the patterns seen in the evolution of the bilateral nervous system is the evolution of cephalization. Cephala always refers to head, and ization refers to some process of formation, so this is the evolution of heads. So cephalization is the concentration of nervous and sensory structures into a head or anterior end. And we're going to see that this is highly correlated to locomotory abilities in animals. Bilateral moving organisms we're going to see are oftentimes on the move. And if you're going to be on the move, you want to go head first into the environment so you can tell what you're getting yourself into. And in some lineages, like vertebrates, there's another evolutionary pattern of greater and greater complexity in the brain. So the evolution of larger brains and increasing brain complexity is a pattern seen. Well, let's review the diversity of sense organs seen in animals. Sense organs are receptors that receive information about the animal's interior or external environment. So they're part of the afferent neurons in the peripheral nervous system. One class of these are called chemoreceptors, likely the oldest sense in the animal kingdom, and it allows what we call chemotaxis, or orientation relative to chemical cues, again in the external environment or the internal environment. So this allows animals to do things like find food by picking up the chemical distribution of things associated with their food, perhaps finding mates using chemicals called pheromones, establishing territory, so a variety of functions. And the main types of chemoreceptors you're familiar with are things like taste buds, and also organs like your nose, which is associated with olfaction or a sense of smell. Another class of sense organs are mechanoreceptors. These are sensing forces like touch, pressure, pain, stretching, sounds, vibrations, sensing the direction of gravity. So associated sensory structures oftentimes are associated with tactile hairs that feel movement or feel pressure. There can also be general receptors deeper in the skin that help to establish different levels of pressure. Here's an example in fish of neuromast cells, which if you look at them up close, they have hair-like triggers on the hair cells. And these are lining the lateral line system of fishes. You can see this inner canal associated with the lateral line system where these sensory nerves are connected to these hair cells. And these canals are picking up changes in water pressure, picking up vibrations in the water, and sometimes even detecting weak electrical signals. Another example of a mechanoreceptor are ears. Ears are associated with hearing, which is picking up vibrations of sound. This is primarily seen in terrestrial vertebrates, and it's not really seen in too many terrestrial invertebrates. However, one example of an invertebrate that does that are moths. Moths have air sacs that can sense vibrations associated with potential predators like bats. So they don't have ears like you're familiar with. Their ears their hearing structures are associated with their thorax, but it's doing the same thing, picking up vibrations associated with sound, and they're using this to detect a potential attacking predator, and they go into these dives to try to escape the attacking bats. Ears are also seen in some spiders and even some crustaceans, but again, it's primarily seen in terrestrial vertebrates. Another type of mechanoreceptors are statocyst and vestibular organs. These are all about sensing equilibrium, figuring out the direction of gravity so you can maintain a balance. So these sense the movement of hard floating objects or internal fluid relative to the position of sensory hair cells again. So here you can see in the head of a crustacean, we have this statolith, which lith refers to a rock-like structure, and stato refers to some equilibrium type mechanism. So this is floating in a fluid, and the direction of gravity is sensed as this sinks and touches specific hair cells. So it tells the direction of gravity. Here we have the inner ear system of a vertebrate, in which in these semicircular canals we have this endolymph, this internal fluid that has suspended small crystals in it, and as those crystals touch these sensory structures linked to hair cells, it again triggers the direction of gravity or the direction of movement. And the final type of sense organ we'll talk about are photoreceptors. And these can be very simple, even seen in protozoans. Single-celled organisms have these organelles that can detect the presence or absence of light called stigma. These organelles are called stigma. But the animals you're most familiar with have more complex organs that allow the formation of visual images. So here we have a picture of a human eye in which we see light comes through the pupil. It's focused through this lens to form an image on the back of the retina, and this is then transferred into electrochemical signals and sent to the brain via the optic nerve. We're going to see many invertebrates have a compound eye in which each of these individual little components of the eye, called ommatidia, can receive just a small fraction of the visual field, but because they have many of these linked together, they're all sending this information to the brain via the optic nerve, and then in the brain all of this information is put together into a single image. So photoreceptors are important in simple animals for simple phototaxis, basically moving to or from an object depending on the concentration of light. So that would be something that this protozoan could do. But more complex photoreceptors that form images can be used to help judge the quality of a mate and use mate choice, make foraging decisions, to hide from predators, or become more efficient as a predator. So in review, nervous systems are all about the movement of electrochemical signals. The structures that transfer this information are called nerves, which are nothing more than bundles of nerve cells, which are called neurons. And we looked at the detailed anatomy of a neuron, starting with the dendrites, which are the receivers of the information, passing through the nucleated cell body, and then the largest part of the neuron is the axon, the long axis. Sometimes this has a insulating myelinated sheath, which helps to speed up the nerve action potential, but most invertebrates don't have this. And the signal ends in the terminals or synaptic knobs, where the information has to be transferred to a neighboring cell membrane, either via, rarely, an electrical synapse, more likely a chemical synapse associated with neurotransmitters. How a nerve action potential works is associated with chemical change from a resting membrane potential, in which there's more chlorine and sodium on the outside of the axon, and more negative large anions and potassium on the inside of the axon membrane. So that's the resting membrane potential. And an action potential starts when we have depolarization, caused by sodium flowing into the interior of the axon, changing the inside from a negative net charge to a positive net charge. And as the sodium flows down the length of the axon, with sodium pumping in, we also see potassium moving on the outside of the axon. That therefore requires for us to regain the resting membrane potential to swap the sodium and the potassium, and that's accomplished by the sodium-potassium pump. And again, the transfer of signals between cells at the terminals or synaptic knobs is either done through an electrical synapse or more typically the chemical synapse via neurotransmitter chemicals. And then we talked about the evolutionary diversity of nervous systems. Some of our simplest animals simply have a nerve net without any central nervous system or brain. But most animals have a bilateral nervous system with a peripheral nervous system and a central nervous system, with the peripheral nervous system being associated with afferent or sensory neurons, which are receiving information about the interior or exterior environment, taking this to the central nervous system, and then the central nervous system sends signals on how to respond to this environment via the efferent or motor neurons. And the central nervous system is composed of the brain, the nerve cord, and the small neurons that make the connections between the peripheral nervous system and the central nervous system, the interneurons. And finally, we talked about the diversity of sense organs in animals, starting with chemoreception, which allows for chemotaxis, responding either positively to chemicals or negatively to chemicals. So you could swim away from certain chemicals or to certain chemicals. That's what chemotaxis is. It's movement. Taxis means movement, movement associated with chemicals. And some organs associated with chemoreception allow sense of smell and taste. Another class of sense organs are mechanoreceptors. These allow for touch, feeling pressure, equilibrium, and hearing. And finally, animals have the ability to judge the amount, direction of light, and in some cases actually form vision. And this is photoreception. And this allows for potential behaviors related to avoiding light or moving toward light, and this is called phototaxis.